This lesson is on the structure of DNA and RNA, and here are your objectives. RNA stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid. And we'll see what that deoxy difference between the two of them means in a moment. Both of them are polymers of nucleotides. So let's take a look at what this means. The ribo in ribonucleic and deoxyribonucleic refers to the sugar ribose. Remember that ribose is a 5-carbon sugar. And we number the carbons 1, 2, 3, 4, and 5. Both ribonucleic acid and deoxyribonucleic acid contain a version of the sugar ribose. The difference is here on carbon 2. In RNA, it's an OH like drawn here. That makes it a ribose sugar. In DNA, the oxygen is removed and it's just a hydrogen. That's the deoxy part. We've removed that oxygen, and that's what we see in DNA. So it's only this difference at this second carbon on the ribose that makes the difference between RNA and DNA. So this is a ribose. To make it into a nucleotide, we need to add a base and a phosphate. So here on carbon 1, we're going to attach a nitrogen-containing base. I'm just going to draw that like this for simplicity here, and we'll take a look in more detail below. And on carbon number 5, we're going to remove the hydroxyl group, and we're going to attach a phosphate. So we're going to attach a phosphate, and I'm just going to label that with a P over here. So now that we have a phosphate, a ribose sugar, either regular ribose in RNA or deoxyribose in DNA, and a nitrogenous base, a nitrogen-containing base, this whole thing now is a nucleotide. And so it's the monomer of which RNA and DNA are made of. To draw this more simply, in general, we're going to draw a pentagon for the ribose or deoxyribose sugar. We're going to draw a rectangle for the base like I've drawn here, and we're going to draw a circle for the phosphate. So we've got our base, we've got our sugar, and we've got our phosphate at the top. Let's take a look at this in a bit more detail with some diagrams from Campbell Biology. Here in the center we see one nucleotide with our sugar, our nitrogenous base, and the phosphate group. On the right-hand side we can see the different kinds of nitrogen-containing bases that this could be. There are two classes, pyrimidines and purines. Purines have 2 rings and comprise adenine and guanine. So A and G, they have 2 rings. Pyrimidines, in contrast, which include cytosine, thymine, and uracil, have only 1 ring in the nitrogenous base. And a 1-ring base will always pair with a 2-ring base, or vice versa. So pyrimidines are always going to pair with purines, and we'll see that in a moment. Now you might ask, well, why are there 2 purines and 3 pyrimidines? Well, it turns out that thymine is only in DNA, while uracil is in RNA. So instead of having thymine in RNA, we have uracil in its place. On the left-hand side here, you can see how the monomers of nucleotides are attached together into a polymer of a nucleic acid. The sugar and phosphate form a kind of backbone, and the nitrogenous bases stick out from that. This could be the structure of RNA, which is generally single-stranded, and if we put another strand here upside down, we could get the double-stranded molecule DNA. Again, the other difference between DNA and RNA that we've already seen is here on carbon number 2. Just a note, by the way, about the numbering. Once we attach a nitrogenous base, which as you can see has carbons in it, in order to keep the carbons on the sugar distinct from the carbons on the nitrogenous base, we add a prime, this little apostrophe. So you can see now that this is called the 5' carbon, and this one is called the 3' carbon. And you're going to hear those terms a lot because they indicate directionality to the strand. If you look over here on the sugar-phosphate backbone, the carbon over here is the 5' carbon, and it's closest to this end, so we call it the 5' end. Here on this end we have the 3' carbon being the closest, so we call this the 3' end. And that's how we tell the direction that the strand is oriented. Before we leave this diagram, we can talk briefly about how the monomers attach to form this polymer, this nucleic acid. So If we have a nucleotide that has two more phosphates attached to it, we call this a nucleoside triphosphate. And this is actually the starting structure for the monomers. This will come in here and add on the 3' end, and we will lose these two phosphates. And actually the splitting of these two phosphates off and then from each other releases energy, which drives the whole process. And again, this diagram's from Campbell Biology. To summarize differences between RNA and DNA then, RNA contains a ribose sugar, whereas DNA contains a deoxyribose sugar. The difference, once again, being on that second carbon. DNA is double-stranded, but RNA is generally single-stranded, although it can bond with itself to form double strands, and sometimes that's important, as we'll see when we talk about transfer RNA, for example. We've seen that DNA has 4 bases, adenine, cytosine, guanine, and thymine, whereas RNA also has 4 bases, but instead of thymine, it has uracil. Let's take a look at the structure of a double-stranded DNA molecule. Here we see the sugar phosphate backbone, and the specific kinds of linkages between the phosphates and the sugars we call a phosphodiester linkage. So this kind of linkage in here is called a phosphodiester linkage. Now, we also have labeled the 5' and the 3' end. They're labeled here for us, and we can see there's the 5' carbon, there's the 3' carbon, and the hydroxyl group coming off of it. Notice that A is always bonding with T, and C is always bonding with G. You might also notice that C and G have 3 hydrogen bonds. These are hydrogen bonds in here. And A and T only bond with 2 hydrogen bonds. And so, in fact, the C and G bonds are more stable than A and T. So if you were to heat up this DNA molecule and provide a lot of thermal energy, you'd disrupt the AT bonds before the CG bonds. Now, as we'll see, the sequence has meaning. The sequence of nucleotides, and specifically the sequence of bases, A, C, G, and T, is going to specify which amino acids ultimately are put into the primary structure of a protein, and therefore ultimately determine the 3-dimensional structure of the protein and its function. So this sequence carries a lot of very important information. Again, notice that A is pairing with T, or in the case of RNA, it would pair with U because there's no thymine in RNA, there's uracil instead, and C pairs with G. Also notice that the 2 strands are pointed in opposite directions. The 3' end of this one is down here, and this is the 5' end. They're upside down relative to each other. We say that they are anti-parallel. One more thing before we leave. If complementary base pairing, this is called complementary base pairing, where A's bond with T's or U's and C pairs with G, if complementary base pairing always occurs, then once we know the sequence of one side of the DNA double helix, we know the sequence of the other. And that becomes extremely important for how DNA replicates and also how DNA leads to the synthesis of RNA, which ultimately leads to the synthesis of protein. We'll explore all of that in upcoming lessons.